Hi everyone, this is a very general overview for your final exam. Again, we're not going to go over any specifics here. This is just meant to be nice, short, and sweet, so it gives you a nice direction of where you should focus your studies. But make sure that you look through your labs, look through your assignments that we've gone over in the last 10 weeks before you go ahead and start the exam. It is timed, so I want you to have all that knowledge in your head instead of trying to look up answers, because you won't have enough time to do it that way. One of the first concepts that you'll need to know is dominant versus recessive alleles. So remembering that capitals are always going to represent the dominant allele and the lowercase letters represent the recessive allele. Dominant is always going to be expressed. If the dominant is present, it's always expressed. That's not true of the recessive alleles. As we know, the recessive genes can be carried without being expressed. But in the rare cases that you have what looks like two lowercases, and the example here is two lowercase f's, if there's no dominant present, then the recessive will be expressed. So make sure you review that if you're unsure. And also make sure you understand the concept of an X-linked allele and what that means, who that affects, and how often it affects which gender. If you're confused about that, look through the announcements. There's 4 pictures a couple weeks back that show how a carrier from a mother or father can affect the children. Another thing that we talked about in the last half of this term was cancers and what malignant versus benign cancers mean and how they grow and what happens. This is a great chart from your book, Table 18.1. It does go over benign versus malignant, what it means, how fast they grow, and what it looks like. So definitely check out this picture. And then at the very bottom of the slide, proto-oncogene versus oncogene. There's a very brief paragraph in your book about that. I encourage you to check out those definitions before you take the exam. Another concept that we went over was genetic drift. Definitely know the definition of genetic drift and also be able to answer these questions. How does it fit in with the allele frequency? And that has a little bit to do with the Punnett squares, a little bit to do with the bunnies that we did in week 9 lab, and just make sure you understand how that works. You'll need to know what natural selection is and how natural selection differs from the definition of evolution. And then, speaking of evolution, understand the difference between a microevolution and macroevolution. And if you just look at the basic definition here, micro means small, macro means big. So on a small and a big scale, what is actually changing through the generations? The last one, the last point here, migration and environmental factors. Another great example for this is the bunnies in week 9, when we pulled the red and white beans out of the bag. Make sure you understand the concepts behind that lab. Alright, GMOs. We should know by now that this stands for genetically modified organism. We talked a lot about genetically modified foods, how that affects our bodies and our environment, how we feed people. Know why it's an important issue and understand the risks involved. Alright, by this point we've talked a lot about Punnett squares and how you inherit genes from your parents. Make sure you know that there are 46 chromosomes, meaning 23 pairs in a human being. That 23rd pair is not always identical. It's not homologous like the first 22, because that 23rd pair determines the gender or sex of the individual. There will be a question about identical versus fraternal twins, so make sure you understand how those two phenomenons happen. When it comes to DNA, you will see these 4 words a lot throughout the test. It helps to know exactly what they mean because it's very common to get these terms mixed up. So Know that a gene is a unit of heredity. A genome means a complete set. So you have a genome that's different than my genome, that's different than your parents' genome, and so forth. A genotype is specific to an individual, and then a phenotype is an observable genotype. So it's like a hair color, eye color, height, weight, that sort of thing. Alright, since we're talking about making babies, understand where the fertilization occurs, and I mean where as in where in the reproductive tract. What obstacles sperm encounter along the way, and then also what fetal development means. When does it start? How long does it take? What major things are happening at certain times? So example, when does the heart start beating? When are the limbs formed? And so forth. Just know the major events. Don't spend a lot of time on that. Just make sure you do refer to it before you start the exam. There are at least 2 questions about transgenic bacteria. I would refer to page 475 for this topic. Make sure you review the purposes of the transgenic bacteria, both good and bad. What are some good things we can do with them, bad things that could happen, and then also the definition of transgenic, just in general. What does that mean, and how do we use that in a laboratory? The carbon footprint, that's one of the last things we talked about in this class. A lot of people are confused about what that means. It doesn't mean just like garbage that you're throwing away. It doesn't mean the exhaust fumes from your car as you're driving down the road. Make sure you understand what types of waste are included in this carbon footprint. And if you haven't gotten to the discussion yet where you need to look up your own, that will make more sense when you get there. Lastly, I wanted to go over Punnett squares one more time. When you're drawing out a Punnett square, remember the upper corner here, this is blank. That doesn't mean anything. We're going to put one parent here. We'll use ours. So we'll make this top parent have one dominant and one recessive allele. And we'll make this other parent have 2 dominant alleles. These 4 boxes are representing the children, and if we carry the capital down, the little r or recessive gene down, and then carry the other parent across, your Punnett square will look like this for these parents. So the children of these 2 parents have a 0% chance of showing or expressing that recessive allele. All 4 of these children will have a dominant allele, and that's what will be expressed. If we change this up, let's say that both parents have a heterozygous allele, we will have a change here in the children. And in this case, this child right here has 2 recessive alleles. So of these 4 children, there is a 25% chance that that recessive allele will be expressed. If you're having any trouble with Punnett squares, let me know. But at this point, I think we've all got a pretty good handle on it. I will also post a video that has to do with the lab portion of your final exams. Make sure you check that out as well. If you have any questions, let me know. Thanks.